Welcome to another episode and another year of the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview authors from our feature articles, the papers of the month, or other articles published in that month's print issue of the Diabetes Journal. We also have a Sweet Talk segment led by Kevin Williams, where we get a little creative in our scientist and topic interviews. My name is Darlene Sandoval, and I'm, as always, joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are you all doing today?
Hey, Dar. How's it going?
Hey, doing pretty good.
So technically, it's still 2025, but when this is aired, it'll be 2026. Can you believe we're entering our third year of doing a podcast?
Yeah, they're starting to really kind of add up, huh?
Yeah. So I thought
when do we become tenured?
Oh, that's a good question. Does that
Is there
mean
any
you get
pension with this?
Yeah, exactly. A pay raise, and
Yeah.
they have to keep us employed forever.
Any residuals, I guess. We're going to get some residuals on the episodes.
All right, so I thought for our banter today, we would talk about hot takes or predictions for 2026. I think we can make these either funny or serious, whichever direction you feel like going. But I thought I'd start with a funny one.
Yeah.
I predict that Dan Drucker will retire from posting about new articles and instead post about Canadian fashion trends.
What is the latest Canadian fashion trend?
Jeans.
What is it?
Jeans.
Oh,
Jeans?
Denim. I'm sorry to all of our Canadian friends.
And down jackets, right?
That's right. That's right.
The other one that I was thinking of is that maybe Randy Silly will finally update his presentation jokes. He's been using the same jokes since I was working with him in Cincinnati.
Yeah, why abandon something that works, right?
That's a very good point, and that's probably what he would say.
Yeah.
It's kind of how I keep track of where we are in the presentation.
That's true.
Be a mile marker here and there. So mine's more general.
Okay.
I think from our friends in the beta cell development world, we're going to remove a gene and induce diabetes in a mouse model. Screw up a beta cell and you get a screwed up beta cell.
Yeah.
And then closer to home, I think we're going to jump straight from like 2 incretin hormones to like all 20.
Yeah.
And I know that there aren't 20, but we're going to find them, daggone
We
it.
are going to find them.
Yeah.
I agree.
We're going to put them all in one molecule somehow, a little super glue.
Yep.
And those are my predictions.
Yep. I like it. Just staple GLP-1 to everything.
Yes. Agonist, antagonist combined for
Yep.
every one of them.
Yep.
It'll start going into beverages. You just start consuming it.
There you go.
I made a joke at a talk once that I'm not sure went over very well with everyone in the crowd, but I said we're going to start putting GLP-1 in the water and we'll fix everything in DC.
Replace the fluoride.
Yeah, exactly.
It'll certainly cut out on government-sponsored dinners.
Yep. That's right. Save us some money.
That's right. I'm all for it.
Yeah.
Do you got any, Kevin?
I guess I went a little bit more on the serious side. I didn't joke around a bunch,
you
but
did.
I was just thinking back at, this last year, there's a term that came up that I had never heard of before, but NAMs, NAMs, and the non-animal models, and the initiative by the NIH to push studies in non-animal models. And we actually talked about this in the November episode with Alex Banks and Corey Holman and Luis Lantier, who all run cores and their heavy interest in trying to understand energy balance and glucose homeostasis. And I think they had a very thoughtful discussion on it. And while definitely the initiative is an important initiative and necessary to try and understand translational physiology from these rodent models to or preclinical models in general up to humans, I think they did provide a good discussion that it's incredibly hard to model these mammalian systems in situ in a dish or a culture. And so it seems kind of far removed to try and focus completely on the NAMs in this field in particular, and trying to be able to understand the physiology and how it all works together as a system when you're talking about these individual cells or culture models.
Yeah. Yeah, as an integrative physiologist, it's hard to imagine we're going to be able to get the information we need from some of these models. But I think, it's all pieces of a puzzle and there is opportunity there, but I do think regulating it we're regulating it a little before we're actually ready for it, right? Where we have the validated tools
Right.
to actually make a difference in human physiology.
I think I would appreciate incentivization before the regulation, right? That
Yeah.
that, but I don't make these decisions, so
Yeah, that is an interesting idea, right? Like provide some innovation awards or something like that. A
yeah,
little
targeted RFAs that
Yep.
just, push people to think about it and apply it and develop those tools. I think you're
Yeah.
exactly right.
Yeah.
Yeah, on it, Kevin. We were having fun, and now we're thinking.
Well, it has, I think, for all of us that touch on both diabetes and obesity, it has been it is a very interesting time in our field, right? And I think the brain is finally getting its um, consideration for how important it is in regulating body weight, which it was always a fight, I think, previously. So I guess that's good.
I was just going to add to that that you're talking about the incredible time we're in, and focus on potentially the involvement of the brain or the advancement of these different scientific inquiries. But one thing that I was also thinking about is the AI and
Yeah.
language models, machine learning, and the advancements that we've seen just over the last. I mean, it's only been a couple of years, probably in the mainstream, that people have started using these machine models or machine learning in order to advance their science. And while I think it's probably far removed from replacing the individual science experiments, because you really do need to understand the fundamental physiology. And if these AI systems are based off of, or the machine learning is based off of incorrect information, you're not going to come to a proper prediction or conclusion. But the augmenting ability of these tools, in the next year, I mean, I can only guess or imagine what advancements could come from that could be related to predicting drug design or predicting side effects, or maybe the physiology that we're actually studying, and maybe moving towards, that NAM kind of investigation, as long as you can truly have a true understanding of the underlying physiology at play. Then there's going to be incredible advances, I think, there in the next few years that we can't even imagine.
Yeah, I remember years and years ago, I was sitting next to somebody who was in pharma, and we were discussing about whether there will be ever a situation where you can use, a computer algorithm to do a glucose clamp, right? And I
Mhm.
Yeah.
feel like I don't know if just there hasn't been, I haven't seen anything like that yet, but it's got to be coming, right? And certainly we in humans, we were able to sort of come up with a if this then that kind of scenario for the clinical research nurses that were working with us. Um, a
But isn't
mouse is
that
harder
kind of
to
what?
clamp than a human, though.
Yeah, isn't that kind of where we're going, or where they're going with the pumps, right? The insulin pump,
Yeah, for
especially
sure.
the
Yeah.
Right.
integrated
Right.
glucose monitoring with insulin, and sometimes even dual hormone, right?
Yeah.
So
Yeah.
you're right, but we're kind of halfway there.
Yeah. Yeah. We
That's a
just,
great point, that those pumps trying to predict the physiology so that they can
yeah.
get ahead of the changes in the blood glucose and really be able to control it in a very precise manner.
Right, right, or at least a safe one.
Yeah.
Yeah. Yeah. Yeah. And it definitely seems like you have to have glucagon on board to if you really want to control glucose homeostasis, then having glucagon on board to prevent the hypoglycemia is very important, right?
Some of us would argue yes.
What about, do you all think that we will get to a point where the masses will accept that obesity is not a choice?
Oh, that's an interesting one for just in the last week or so, because what is it? The World Health Organization has seemed to come out with recommendations for these incretin-based drugs and pharmaceuticals in the treatment of obesity, really trying to really getting to a point where they're recognizing obesity as a disease.
Yeah.
And there's been a lot of publicity about this just in the last week, and the lack of some of these organizations to even appreciate bariatric surgery as an intervention for obesity early on and even into recent days. But the changing winds of some of these organizations to acknowledge obesity as a disease, it does seem like it might push what you're saying as like the mainstream understanding that it's not a choice.
I think this is really tied a lot to, first of all, we now have treatments, not just one,
Yeah.
but multiple. They seem to be very efficacious for lots of people. And then it comes down to, can we get them to those people, right? The
Yeah.
insurance and regulatory bodies are really the linchpin, I think, right now, because once we treat lots and lots of people, right, it's going to be like having high blood pressure, where you say, well, sorry, I predict it'll be like having high blood pressure, where of course it's a disease and it's a treatable disease, and it's no big deal, right?
Yeah.
Um, but I think you're right. It's going to take those bodies plus maybe the ability to pay for it to really get us there.
Yeah. Also, its impact on the comorbidities might have a bigger impact in terms of being able to get it to more people or seeing that it's important to get it to more people.
Yeah. Anecdotally, I've heard so many times how it's affected people's how they feel. And that's such a hard thing to score.
Yeah.
And I think going back to like, how do you argue to pay for something where someone's like, I feel so much better, which is
Yeah.
probably true and maybe the most valuable side effect of the weight loss, right?
Yeah.
Um, but but again, how do you score that? How do you quantify it? How do you make it something to put in an actuarial table?
Yeah. Yeah, and I imagine you're talking more about sort of energy levels, et cetera, but there was a whole session at Obesity Society this year about food noise, right?
Mhm.
Which is a new endpoint and sort of that drive, that interference of your thought processes because you're always thinking about, okay, when am I going to eat again? There's cookies in the break room and I really want one, but I
Yeah.
shouldn't have one kind of thing. Right. And I have definitely heard that anecdotally that it makes people feel so much more productive because they're not preoccupied by other things. Um, but again, like just like what you said, they're still struggling with how do you measure food noise? And as soon as you describe it to somebody, then they're like, oh yeah, I have that.
Yeah.
Right. And so it's still, we're still beginning to understand that. And these drugs are why we are even thinking about it. Right.
Yes. Yes. Again, without the ability to treat, right? We're in a different discussion altogether.
Yeah. Yeah.
It also raises a future area of investigation that, these drugs, as we're talking about, are highly effective at weight loss right now for a good number of people. Not everybody, but a good
Yeah.
number of people have effective weight loss on this. Not everybody is able to stay on these medications for life. And that could be a number of reasons, whether that's the side effects of the medications, the cost, the accessibility, insurance, all these things. And so I wonder, I have to think in the coming years that a major aspect of research will be trying to understand weight regain after being on these therapies and whether or not there's anything outside of these therapies, which, obviously they're not continuing to take it for whatever reason that we mentioned earlier, if there's something else that can be done in order to sustain the weight loss after it's already occurred. And so I think that could be a very interesting area of investigation that maybe we'll start to see more and more of in the next year, couple years, few years from now.
You
Yeah,
know,
for sure.
I've heard it put forth that maybe some of these older small molecule weight loss drugs might be useful once you lose the large bolus of weight, because the pressure won't be quite as great to lose weight, but to maintain. And they're cheaper, infinitely more cheap. Or sorry,
Yeah,
less
yeah.
expensive. And I guess that frees up the market a little bit too for more people to be treated.
Yeah, yeah, for sure. I still think that it's fascinating to, and I know this is a little bit idealistic, but if we could understand if, like, for example, right, you have that variability in response to bariatric surgery as well. Are those same people that aren't losing weight with surgery the same people that wouldn't lose weight with the agonists, or are these completely different populations? Can
Yeah.
we screen them out? Can we figure out who would be successful, right? Especially when we're talking about surgery, because that is a more drastic solution. And so making sure the right people are getting surgery and the right people are going to medications would be really cool.
I think first we'd have to figure out exactly how the surgery works, right?
Yeah,
Sure.
that's true.
A little plug for some of the work already being done in the room.
Yes, exactly. All right. Well, let's get to some science then, or some more science. Kirk, why don't you tell us about our January paper of the month discussion?
Yeah, so for January, the paper of the month was Killing of Human Beta Cells by CD8 positive T cells triggers inflammatory paracrine signaling and neighboring beta cell dysfunction. To discuss this paper, we were joined by the first and corresponding author, Masaya Oshima, who is a contract researcher at Institut Cochin in Paris, France.
All right, Kevin, what do we have for our sweet talk segment?
Yeah, today we're focused on stress and its effects on blood glucose regulation. We're asking questions like, why does stress send blood glucose through the roof? Which brain circuits are actually controlling these responses? And if stress-induced hyperglycemia is always harmful, or could it possibly serve as an important physiological purpose? Our guest today has dedicated her research to answering those exact questions. It's Dr. Sarah Stanley, who is an associate professor at the Icahn School of Medicine at Mount Sinai in New York, and she is a recipient of the American Diabetes Association Pathway to Stop Diabetes Award. I think she brings an excellent, wonderful perspective to this conversation, so stick around and find out more.
Awesome. Well, I just want to, since we're starting our third year, just want to thank you both for hanging out with me on occasion to do these recordings. It's been so much fun, and looking forward to another year. So, with that, let's jump into the episode. Today, we will be discussing January's paper of the month titled Killing of Human Beta Cells by CD8 positive T cells triggers inflammatory paracrine signaling and neighboring beta cell dysfunction. We are joined by first and corresponding author Masaya Oshima, who is contract researcher at Institut Cochin in Paris, France. Welcome, and thank you for sharing your work with us today.
Hello, thank you for having me.
Dr. Oshima, we like to start with an icebreaker, and we haven't provided this question for you. So I was wondering, maybe you can share a fun fact about yourself, maybe something you do outside of science that would give our listeners a little insight.
So I'm a Japanese researcher, but I was born in France and I grew up there. So I'm like a Japanese outside, but inside is completely French.
All all right. So I guess as a quick follow-up, what's your favorite comfort food?
My favorite comfort food. So I said I'm French inside, but my comfort food is Japanese food and mostly ramen.
All right. Very cool. Very cool.
All right. First, we love to give our authors the opportunity to acknowledge any other authors or collaborators on this work.
Absolutely. So I'd like to acknowledge all the co-authors or who contributed to the work and the experimental design. And of course, I would like to thank our funding and sources and sponsors who made it possible to carry out that project and using advanced technologies for that specific publication.
Nice. Yes, always important to acknowledge those funders. All right. And then can you give us an overview of your research program, and then not necessarily the rationale for this particular project, but maybe how your research program kind of led to this project?
So my research focuses on understanding what a pancreatic beta cell is and how it maintains its identity and function, and why it becomes dysfunctional in diseases. And we approach this from the perspective of fundamental biology, developmental biology, but also in the context of diseases where the beta cells are progressively targeted and stressed. We try to modelize the beta cell physiopathological background in type 1 diabetes, type 2 diabetes, but also in other rare disease like monogenic diabetes, but also insulinomas. And to go to our publication, we focus on type 1 diabetes because it's a unique disease where beta cells are not only dysfunctional, but they are also actively targeted and killed by the immune system. And we lack a good and robust model to understand and to study what happens during autoimmune attacks. So we need, especially in a dynamic way, in vitro tool to better characterize the disease.
Nice.
So Masaya, let's I guess hone in a little bit more specifically on this project. In the rationale, you discussed the progression of type 1 diabetes and the promise of using anti-CD3 antibody. For example, I'm going to mess this one up. Teplizumab. Is that right?
Teplizumab.
Teplizumab,
Yeah,
thank you.
teplizumab.
And how you might be able to delay type 1, by inhibiting CD3 activity. How did this work lead to the rationale for the specific manuscript?
So teplizumab has shown that targeting the immune system can delay the onset of type 1 diabetes, which is a major breakthrough. But it's also clear that this effect is transient because it doesn't stop the disease progression. So this tells us two important things. One is the immune system matters, but is not fully controlled by the current interventions. So we still don't understand enough about the state of human beta cells and during the disease progression. So what are the surviving cells learning or adapting? And could understanding that biology help us design more durable and complementary interventions on type 1 diabetes?
Got it.
Awesome. Let's get a little bit more into the methodology of the paper. And you kind of touched on this a little bit, but you use some special cell systems in this project to try to study this immune and beta cell interaction. So can you maybe explain some of these cell systems, maybe more angled towards a scientist that's not necessarily involved in these in vitro studies for type 1 diabetes?
Okay, so we worked with human beta cell lines that was developed in the Schaffman lab, which are called endo-CBH1 cells. And these cells are human beta cells that express insulin and secrete insulin in response to glucose and secretagogues. And what's interesting is that these cells also present at its surface through MHC class 1 molecule as insulin and insulin peptides. So we made these cell lines into pseudo islets in 3D using specific techniques, and we engineered two populations. So we needed these two populations to track both direct killing and indirect consequences on the beta cells that are not directly targeted and not yet been eliminated. So on one side, we have the wild type beta cells that express MHC class 1, and therefore they are visible by the CD8 T cells. And we have a B2M knockout beta cells, and B2M is a component of the MHC class 1. So without B2M, do not have expression of MHC class 1 at the surface, and therefore they are invisible to CD8 T cells. And we use engineered human T cells that we call transductant that express a T cell receptors that specifically recognize a beta cell antigen. So we mix everything together, and in that setting, the T cells kill the wild type cells through classical perforin granzyme A mechanism, but they can't kill the B2M knockout cells because they don't recognize them, because they do not present MHC class 1, and there is no interaction between CD8 T cells and the B2M KO cells. So this setup allows us to get two information, the killing and the trans-actin effect on the beta cells. So within the same well, we can see both mechanisms.
That was great. Thank you.
So I guess I have a quick follow-up there. These, you say these are pseudo-islets. Are they beta cell specific pseudo-islets, or do they also express other endocrine cells?
So these cell lines are human beta cell lines, and they mostly express insulin.
Okay.
And yeah, so it's a beta cell line. So it's only insulin expressing pseudo islets. Gotcha. And the idea would be to use either IPS derived material or ES derived material to get more alpha cells and delta cells together in that setting, or primary islets, or even the idea is to get new cell lines that are alpha cell lines and delta cell lines from human material.
Yeah, very, very cool. So I wonder if you can take us a little bit deeper into this co-culturing system, and how do you validate this model?
So to validate the model, we confirmed that the specific killing of the MHC class 1 was specific by using insulin KO cells and B2M KO cells. So without insulin and without B2M, there were no killing of the beta cells. And when we use a CD8 transductant that do not recognize the insulin peptide, but CMV peptide, there is no killing of the wild type beta cells. So that's the validation of our co-culture system with pseudo islets and transductant. And we also confirmed robust T cell activation markers and a cytokine secretion in this assay to validate the T cells that are activated and secreting normally.
Yeah, very, very cool.
So then you observed that the impact of this co-culture system on beta cell function depended on whether the beta cells were in contact with the T cells. So you saw different findings between those cells that were contacted and not contacted. Can you talk a little more about these findings and the implications?
Exactly. So this was a key discovery, and beta cells that are directly recognized by T cells are rapidly killed by the perforin granzyme system, which is expected. But the non-recognized beta cells, these B2M KO cells that should be safe, are not killed, but they are undergo a profound functional changes due to the inflammatory environment that was spread by the T cells, but also by the beta cells themselves. So we see signs of inflammation in the remaining B2MKO cells. And we also observed a reduced global protein translation that happens inside these cells. And we look at the proinsulin processing, and it's also reduced. Interestingly, we don't see at that specific timing of the experiment a decrease in insulin secretion, but we see some functional changes based on inflammation on one side and on the other side, the protein translation. So it's not just a binary states of the beta cell where it's alive versus dead, but it's also like a in-between state where the beta cells are starting to get dysfunction, and one of these dysfunction is inflammation for sure, and the other interesting one is decrease of protein translation program.
So I guess, can I ask, you're seeing direct destruction of beta cells as well as a secondary dysfunction of those cells that weren't even touched directly by the autoimmune attack. Is that correct?
Yes.
Very cool. Very cool. Sorry, I'm a slow physiologist, so you have to give me a little grace. I'm hoping maybe you can now circle back to some critical takeaways. What are the implications of your work, and where the future research may be taking us? And I know we kind of hinted at that, but maybe you can expand upon it.
Using our model and what we have in the literature right now is that we need therapies that protect beta cell function, not only blocking immune system and immune killing, but we need to protect the beta cell itself. Because we have surviving cells that are not yet destroyed, because the type 1 diabetes is a slow and progressive disease. So we need to be able to understand and to characterize and to protect these beta cells. We need to find out new biomarkers to find when the beta cells are destroyed, and this will lead to a therapeutical window where we can aim for specific disease and disease intervention. And we need to stratify all the diseases using these biomarkers. And using our model, it's quite easy to screen molecules, and we can test immunomodulatory compounds, and we can also test beta cell protective agents, and we can also look for specific compounds that will trigger protein translation of the beta cells. And the interesting fact of our model is it's quite versatile, and we can add different other cell lines. So we can also other cell system. We can add, as we discussed at the beginning, alpha cells and delta cells, but we can also add other immune components such as macrophages, NK cells, to have a more complex immune environment and to understand what's happening. And that will be used as an in vitro mimicking of what's happening in type 1 diabetes environment, and to find cures and new compounds that will slow or stop the disease.
Yeah, very, very cool.
Yeah, really exciting. We want to thank you for joining us today and sharing your expertise and discussing your recent work on the interactions of CD8 T cells and beta cell dysfunction. I encourage the listeners to read the full article in the January issue of Diabetes. Thank you, Dr. Oshima.
Thank you.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and I'm joined by one of our co-hosts, Dr. Darlene Sandoval. Our guest today is Dr. Sarah Stanley. She's an ADA Pathway to Stop Diabetes awardee and an associate professor at the Icahn School of Medicine at Mount Sinai in New York. Sarah is one of the leading scientists trying to understand why stress sends blood glucose soaring and what circuits in the brain are actually controlling that response. Dr. Stanley, thank you for joining us today.
Thank you so much for having me. I'm looking forward to our chat.
We usually like to start off with a little bit of an icebreaker question. And the question I have for you today is, if you could have a chance to meet and talk with any historical figure in your field, who would it be and why?
So I think I'd really like to meet Claude Bernard for obvious reasons.
Good one.
So
Nice. Yeah.
thank you. So not just because he's obviously the father of understanding pancreatic function, gastric function, bile function, and particularly neural control of liver glucose production, but I think also he was a sort of foundational figure in experimental medicine more generally, and also in physiology and homeostasis. So I think he would be a really great person to be able to chat to and learn from.
That's nice.
Cool.
I've never actually done this. I was just thinking about this right now. They have these trees of scientific lineage that you can search online. There's one with like chemistry, there's one with neurosciences. I wonder if Claude Bernard's on there and talking about his potential trainees and lineage that might be in the scientific field today. I don't know, but it would be an interesting thing to kind of investigate, and the possibility that you could be chatting with somebody that has a lineage that goes back to Claude Bernard, which would be kind of cool.
That would be fascinating. I think he's underappreciated compared to some other people like Pasteur. So I think it's time that we promoted him a little more.
Definitely. Very cool. So to start off the scientific chat here, I wanted to maybe define a little bit of the word stress. You know, when most people think of stress, they're probably thinking about some kind of psychological stress, deadlines, difficult relationships, emotional overload. But, you know, stress can include physiological internal stress signals that you talk about in your article that you've contributed into the current issue of Diabetes. And this can include inflammation, blood volume changes, nutrient deprivation, you know, maybe something that we do on a pseudo-regular basis like exercise or some other stressors. And so I was wondering if you could help the audience and define stress from that physiological and metabolic perspective that people might not commonly think about.
I think that's actually a really challenging question to answer because it often depends on the context and also the intensity and duration of the stimulus. But perhaps one way of thinking about it would be a stimulus or a state that increases energy demands and perhaps beyond what the reserves are. I would think that's possibly one definition. So physiological and metabolic stress, I think, are disrupting homeostasis. And as you mentioned, many of those are things like infection, trauma, inflammation, and they are activating the hypothalamic pituitary adrenal axis and the sympathetic nervous system, leading to this altered glucose metabolism and obviously many other processes as well. So I think that would be how I define it, as something that disturbs homeostasis and places an energy demand on the body.
I'm curious with that definition of the use of the word stress. I'm wondering if it's just kind of overutilized in these situations and possibly defining it as a strain on the system, as you were discussing, or some other terminology is more appropriate and might actually help advance how people are thinking about these different paradigms and what they might mean for physiology, and possibly thinking about more of a more specific word or something directed towards understanding these different physiological conditions might advance how we think about it and how we study it.
I think that's very interesting. And I think also there's not a lot of consensus across fields. So what physiologists might call stress may be the response to a stressor, whereas a psychologist might call the stress the initiator. So I think perhaps being a little bit more precise in what we're measuring and what we're looking at would definitely be helpful as we try and integrate across the fields.
Let's dig in a little deeper. One of the things that I really enjoyed about the article that you wrote for this month's issue of Diabetes is how I feel like the perspective when people have written about CNS regulation of stress before has always been, again, like you said, from their very professional perspective, right? If you're a physiologist versus a psychologist, but also from their brain region of interest. And what I liked about your article is really talking about the circuits and how one area of the brain is talking to different areas of the brain, and how that could consequently lead to HPA and sympathetic increases. So maybe at a high level, can you talk a little bit about those sort of overlapping and potentially distinct circuits depending on what type of stress we're looking at or what the brain is responding to?
Yes, I think it's very interesting. And I think the fact that there are so many parallel circuits which have similar functions perhaps suggests that this is a really important physiological response and evolutionarily conserved. We know that it's in multiple species and vertebrate families, so I think it talks to its importance. But looking at which brain regions are involved in different kinds of stresses, we can see that there are some common regions and that there are some regions that are sort of stress specific. I think the common regions include areas such as hypothalamic regions, obviously the paraventricular hypothalamus and the HPA axis stimulation there. Also, a lot of amygdala regions are also common across stresses, and they're downstream circuits which can be very varied. I think one of the interesting things is that there are some cortical regions which are also common across stresses. which are less well understood. But I think the outflows are often very similar. So there's an increase, as we said, in the sympathetic nervous system and in the activation of the HPA axis. What we don't know is whether those stressors also have the same cell bodies, cell types in those brain regions, whether they have the same ensembles, whether the timing of their activity is the same, because we're usually using things like FOSS or trap mice to try and identify the activated regions. And I think those are going to be very important questions as we sort of delve deeper into the circuits.
Yeah. Since you brought up the FOSS trapping, let's talk about some of the strategies that maybe you've personally utilized or that you've seen utilized that you think will really help us understand more about the circuitry behind stress responses.
So I think a lot of what we know about which brain regions are activated, it comes from FOSS studies, as we've said. The initial ones were more focused on particular brain regions, depending on what your interest is. And I know we've done that for the medial amygdala. There have been more broad, unbiased studies as well, using serial sections and FOSS. And I think now having tissue clearing and being able to look at FOSS across multiple brain regions and analyze those in depth, either with FOSS staining or with the TRAP2 mice driving a fluorescent reporter in response to neural activity, we're getting a much broader view of brain regions that are involved with different stress responses, and that's helping us pull out regions that we wouldn't necessarily have thought of as being part of the stress response. So that's one tool which I think is very important, having the ability to image the entire brain and analyze that in depth, looking for the connectivity between regions, and also looking for regions that have their activity down-regulated in response to stress, which we haven't really focused on at all. I think it's going to be interesting. And then that obviously doesn't tell us about the temporal dynamics. So I think we have to combine that with more functional imaging tools, whether that's fiber photometry or miniscopes. But again, then you're sort of limited to specific brain regions. It's hard to do that across broad swathes of the brain.
Yeah.
And it's always important to assess function as well. We don't know whether these are in response to the stress changes in corticosteroid, glucose, heart rate, etc., or whether they're actually driving them. So I think the functional assessment becomes a crucial part.
Yeah, that's great.
I had kind of a question that was building on that, or kind of analogous, and looking at some things that you kind of alluded to in that previous answer, and whether these stress responsive neurons are simply sensing these changes in stress, or are they sensing changes in the blood glucose levels that could be occurring under these stress conditions, whether the neurons are actively driving those changes in physiology, the changes in blood glucose, or maybe the stress hormones or what have you. In other words, who is actually in the driver's seat, and what do we know about that temporal regulation, the timing of neural activity relative to the rise in blood glucose levels, or the stress response for hyperglycemia? And maybe some recent literature that has reevaluated that type of information as it relates to some of the strategies you mentioned with FOSS-TRAP or CFOS mapping, or using fiber photometry, or these different methods. Is there some new information that's been gathered from these different technologies on being able to define those temporal regulations?
I think, as we mentioned before, the calcium imaging studies have been really helpful, combined with the peripheral readouts at the same time. So if we combine the fiber photometry with continuous glucose monitoring, we can look at the temporal dynamics in much more detail. Or if we can combine it with heart rate monitoring at the same time, we can also see whether the activity in the neurons precedes the physiological changes. Because a lot of, certainly for blood glucose, a lot of the changes that we see in terms of activity are also in brain regions where we know that there are glucose sensing neurons. So it becomes really challenging from the FOSS and the TRAP studies to tell whether or not they are responses to changes in blood glucose or driving them. But I think the ultimate test is also the functional studies that we just discussed. If we can actually go down the activity and see that that has an effect on the physiological readouts, whether that's blood glucose or heart rate or appetite, which is also influenced by stress, or behavior as well, which is obviously a huge part of the stress response. So I think it's going to be a combination of multiple technologies and tools and readouts that's going to help us try and dissect the contribution of each brain region and cell population there, and even cell population specific circuits.
Yeah, I think it's a really important point. The in vivo imaging strategies and different types of strategies to record the activity of cells in real time has really advanced a lot of our field, right? Like you can think back to just, I don't know, within the last 10 years or so in understanding feeding behavior and not really appreciating how rapidly some of these neurons are changing their activity, either being stimulated or inhibited or what have you in response to food presentation or actually ingestion of food and what's happening in these states of glycemia that could be changing in response to stress or in response to these stress hormones itself. So I think it's a really cool future direction, the area of investigation for this field.
I completely agree. And I think, as you were mentioning the feeding circuits, one thing that's been very interesting is to see how much the sensory circuits are feeding into the activity of those feeding circuits. And I'm sure that's probably going to be true for many of the stress circuits as well, because they're integrating visual, olfactory, somatosensory information about the animals surrounding as well as the internal milieu.
So a major, I think, endpoint that you're interested in, and certainly the focus of the article that we're discussing today, is this stress hyperglycemia, right? And so tell us a little bit about, it seems you're coming at this from the perspective of the stress hyperglycemia being dysfunctional. And so is it always dysfunctional, or is there such a thing as a normal increase in glucose in response to stress that might be an important physiological response? Or does it become more of a problem for the diabetic patient that, just doesn't have as much acute control over their glucose levels?
I think I would probably say it's a physiological response under most circumstances, and that is based on the clinical data from patients who are hospitalized. If you look at the stress hyperglycemia ratio and compare their preceding blood glucose, which has been calculated from their HbA1c, with the blood glucose at admission, you can see that when those are quite closely aligned and the ratio is around about 1, they have much better outcomes than individuals who have either an exaggerated or an impaired glucose response to stress. So that to me suggests that actually having an adequate response to stress in terms of the glucose is actually very important functionally and physically.
Yeah.
And that may be one of the reasons why there are so many pathways, as we mentioned, that are driving this glucose response. So I think I would say that under most circumstances, it's physiological. But as you mentioned, if it's exaggerated, whether that's because there isn't the insulin to try and downregulate it, or whether it's impaired because of adrenal malfunction, for example, or adrenal inadequacy, then it does seem to have adverse effects, not just acutely within the hospitalization period, but also for months and maybe even a couple of years after that hospitalization period.
Yeah. It makes me think about anxiety. So, anxiety is sort of, you might have a physical symptom and then your brain turns on, right? And you're sort of like, oh, what's wrong with my heart rate? And then that like makes your heart rate, right? And it's sort of this vicious
Exactly.
cycle.
Yeah.
And you could imagine that the same thing could potentially happen with glucose, where maybe it's slight, your glucose response might be slightly inappropriate in response to stress. And then your stress response drives it even further, and it becomes sort of a vicious cycle.
I mean, in PTSD, for example, which is
Yeah.
the sort of behavioral or psychological parallel of that, they have an increased risk of type 2 diabetes and cardiovascular disease and many metabolic dysfunctions. So I can see that that is indeed a vicious circle and that they're very closely related.
Yeah.
And another question I had is related to trying to link preclinical to clinical studies and observations of stress and what that's doing to glucose levels. And there's always a challenge, I guess, on both sides, trying to make the translational relevance from these preclinical models into the clinic and having potentially clinicians understand the utility of these preclinical models and describing potentially what's going on in their patients. And in preclinical models, for these stress conditions, they're using a variety of different types of challenges, anything from restraint stress, predator odor, lithium chloride injections, maybe doing hyperglycemic or glycemic changes in order to elicit some kind of a response. And I was curious if you could talk about, when you're thinking about your own studies of the translational utility of these different types of preclinical models to model stress and what that means for understanding or treating the glucose dysregulation that could be occurring in the clinic.
So I think in the preclinical studies, many of us are trying to isolate particular modalities. Is it a visual stressor, an olfactory stressor? a physical stressor, the restraint, and to try and narrow the circuits that we're stimulating with each one to see if we can sort of parse apart which stimuli are acting on the population or the circuit that we're looking at. So that has advantages, obviously, because we are going narrower and narrower, and hopefully can find areas which are switched on by one stimulus but not the other. But as you mentioned, this doesn't seem to have much relationship to clinical and let alone day-to-day life, where stresses are definitely multimodal. So I think there is probably a middle ground in terms of translational studies where we can try and find a stressor which we can use in a preclinical model, an imaging modality with functional MRI or FDG PET scanning, where we can look at brain activity in translational studies that's then relevant to more clinical populations. So for example, LPS, we can use in mice as an inflammatory stimulus, if you like, and then we can have sort of an inflammatory stimulus possibly in humans, maybe. The other thing I've seen is thermal stimulus as well that's been used in imaging studies, which we could definitely use in preclinical, and perhaps in the time of climate change, that's also going to become an issue for human populations as well.
All right, so let's go more broad now and maybe talk a little bit about the actual ADA pathway to stop diabetes award, and do you have any thoughts about how it shaped your lab's trajectory and allowed you to pursue questions? And maybe do you have any advice for folks that are maybe thinking about applying this year? And
So I would definitely say that it was transformative.
yeah,
It was right at the beginning of starting my independent lab, and not only was it a substantial financial investment, but it also came with a lot of flexibility to allow us to pursue these studies. It wasn't linked necessarily to showing that we'd made progress on the aims that we proposed, but allowed us to follow the science, which I think was really
yeah,
important.
yeah,
And a lot of funding now doesn't allow that. Some are more restricted than others, but I think it was really important, particularly at that stage where we were moving into a relatively new area of how stress impacts glucose control, which we hadn't done before. So this was more outside the usual sort of homeostatic regulation in the hypothalamic regions. We were going to a new brain region, which we didn't know as well. We're looking at stress parameters, which we didn't know as well. It really allowed that to be developed in the lab. So
yeah.
it was completely transformative. Yeah.
Do you find that it allowed you to take some technical risks that you may not have been able to take otherwise?
Yes, I think that's
Yeah.
absolutely true. I know we were able to set up the fiber photometry, which we hadn't got in the lab. I'd used optogenetics with other people's setup, but we also allowed us to have our own, which meant that we could do many more studies and explore other brain regions that we wouldn't probably have been able to do because we'd have a limited time on
Yeah.
other people's equipment. So that was one aspect. So obviously the financial aspect was hugely important. I think the other thing that's incredibly important is the mentoring and the community for the Pathway
Yeah.
Award.
Yeah.
So meeting with the different mentors and people who are giants in the field and being able to get their point of view after you've done your presentations at the ADA and feedback on the work and their suggestions about next steps or alternative hypotheses was just hugely important. And that was also true of the other individuals who got the Pathway Awards. You saw
Yeah.
a whole different aspect to their science, which allowed you to integrate technologies that they used and different aspects of their work. Whether if that was a clinical aspect that made you think about how this could impact the circuit that you were working on. And I think those were incredibly important.
What advice do you have for junior investigators thinking about applying for this award?
I think it's much more competitive now than it was.
Yeah.
I think just the funding environment altogether is more competitive, and
Yeah.
that's reflected in the Pathway Award as well. But I think if you can apply, I would definitely do so and get as much advice and support from other individuals who work in your field as possible, whether that's inside your institution or outside. I think people are always happy to provide feedback and advice.
Yeah.
And that's always been helpful for me. Yes, but I would definitely encourage anybody who's thinking of it to apply.
I loved you touching on the mentorship component of that award. It is an unusual aspect of the award, but so beneficial to the awardees, being able to present, as you mentioned, to those leaders in the field and getting their feedback. And it's really an investment by the ADA, not only with the money, but also the effort and time of these individuals, and committed to the development and growth of the awardees. So I think that's really a cool aspect of the award, and it's cool to see the fruit of all of that with awardees like yourself, and the work that you've been able to do.
I would absolutely agree with that. I think that's as well, money's always important because you have to support your lab, but it's definitely a huge, huge benefit to the Pathway Award.
Thank you, Dr. Stanley, for joining us and sharing your insights into neural mechanisms regulating blood glucose control. I encourage our listeners to read her article in this month's issue of Diabetes. It's titled Neural Regulation of Blood Glucose in Acute Stress. Dr. Stanley, before we go, could you let our listeners know where they can follow your work? Do you have a website, a lab page, or maybe you are on the social media sites?
Yeah, you can definitely find me on LinkedIn. Our lab website is also available through Icahn School of Medicine, and we tend to update that and put our latest publications on there, and you'll read about the amazing progress of the people in the lab, who are the most important part, I think.
That's great. A really wonderful discussion. And as always, don't forget to like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.